Hello students, today we are going to discuss the topic of histology of endocrine glands. I know histology, all of you are not studying, you are not referring to the textbooks also. So I will not make this lecture a detailed one, but I will keep it short and we will discuss only the important things in the histology slides that are important from theory point of view, as well as we will discuss few important viva questions. In this lecture, we will discuss the important slides of the endocrine system. We will discuss in detail about the common slides which have been frequently asked, and we will rapidly cover the uncommon slides. We will only see how to draw sessions of important slides, and rest of the slides I will share the diagrams. So let us go to our presentation. So today's topic is endocrine glands. So what are endocrine glands? These are ductless glands. We have to differentiate in between endocrine and exocrine. This is an important viva question. So this question, it will be frequently asked in your viva exams. What is the difference between exocrine and endocrine glands? So exocrine glands, they have ducts, whereas endocrine glands, these are ductless glands. They exist either as a discrete organs. So the discrete organs we are going to discuss today, or they will be found as small clusters of endocrine cells in different organs. Like, so first we will see the discrete endocrine glands. We will enumerate the discrete endocrine glands first. First and foremost, it is the pituitary gland, which is also known as hypophysis cerebri. Next is the thyroid gland. Then we have parathyroid gland. It is a smaller gland situated on the posterior aspect of the thyroid gland. Then we have adrenal gland, which is also known as suprarenal gland. And next we have pineal gland, which is again a part of central nervous system. The endocrine tissues, which are found as a clusters with the other organs, these are islets of Langerhans in the pancreas, interstitial cells of Leydig in the testes, then ovarian follicle and the corpus luteum. Other examples, these are chorion in placenta, Lassis cells and J.G. cells in kidney, and enteroendocrine cells which are found in the GIT. So whenever you are writing a short note of the endocrine glands, you should remember that there is a basic structure or there is a basic formula to write short note of endocrine glands. You should follow the same fashion, that is stroma, parenchyma and capillaries. So at least you should write this, that there is a stroma, there is parenchyma and there are capillaries, because there will be a supportive tissue which is called as stroma. Then there are parenchyma formed by the secretory cells. The secretory cells are supported by the stroma, and the secretions whatever secreted by the parenchyma, they need to be poured into the circulation, so there has to be abundant capillaries. Basic structure of endocrine gland, it is parenchyma, which composed of secretory cells, and stroma, which consists of connective tissue, and numerous capillaries. First we will discuss pituitary or hypophysis cerebri. We will not go into the detail of the gross anatomy because we have to keep the topic short and sweet. So this is the diagram you already know of the gross anatomy of the pituitary gland. It has got pars anterior, pars posterior, in between there is pars intermedia, and it is attached to the base of the brain, and it is in continuation with the hypothalamus. So its stalk. So in your short note of the pituitary gland, this word you should mention, that is hypothalamo-hypophyseal portal system. So it has to be there. So hypothalamo-hypophyseal portal system, it is system of capillaries present in between the hypothalamus and anterior lobe, or the pars anterior, or we can say adenohypophysis of the pituitary. So cells are arranged as irregular cords or clusters which are separated by the capillary plexus, or we can say in other words, the cells are arranged in between the capillary plexus. So what are the cells? Cells are basically chromophils and chromophobes. Chromophils, these are darkly staining secretory granules. They are having darkly staining secretory granules, and these are named as chromophils because they are having affinity for the stains. Whereas chromophobes, these are poorly staining because they are having phobia of the stains. That is why they are called as chromophobes. These are basic questions asked in the histology viva. What do you understand by chromophils and chromophobes? Chromophils are having cells having affinity for the stains, and chromophobes, these are the cells having phobia of the stains. So the cells chromophils and chromophobes. So again, we will discuss the important question, that is why these are called as chromophils, because they are having affinity for the stain, and chromophobes, these are having phobia of the stains. Chromophils are further classified as acidophils and basophils. The acidophils, these are having affinity for the acidic stains, whereas basophils, these are having affinity for the basic stains. We know that acidophil, acidic stains, so they will be seen as pink color, and basophils, they will be seen as a blue color. So hematoxylin and eosin. So acidophils, they will stain with the eosin, whereas the basophils, they will stain with the basic stain, that is hematoxylin. There are two stains, hematoxylin and eosin. Eosin is pink in color, whereas hematoxylin stains the structures in blue color. The acidophils and basophils are further classified according to their secretions. So they produce certain hormones. We'll see the next slide. Chromophils, these are classified as acidophils and basophils. The acidophils, these are having somatotropes responsible for secretion of growth hormone, and lactotropes responsible for secretion of prolactin. Next we have basophils. The basophils, they are secreting gonadotropes, that is FSH and LH, follicle stimulating hormone and luteinizing hormone, whereas thyrotropes, these are secreting thyroid stimulating hormone TSH, and corticotropes, these are secreting adrenocorticotropic hormone. Pars intermedia, it is an interesting region of the pituitary gland. It is very characteristic. So let us see what is the characteristic about the pars intermedia. Pars intermedia, it shows colloid filled cyst or follicles which are lined by basophilic cells. Okay. So if you get a question in your viva, what are the other endocrine gland apart from thyroid which shows colloid filled cyst, your answer should be pars intermedia of the pituitary gland. What does pars intermedia secretes? The pars intermedia, it produces melanocyte stimulating hormone. So we have seen in the beginning the hypothalamo-hypophyseal portal system. So let us see it in detail what exactly it is. So what is a portal system? Portal system, it is defined as, it is a system of blood vessels having two sets of capillaries. So it is a vascular system present in between two sets of capillaries. So here in the pituitary gland, the first set of capillary, it is located in the hypothalamus, that is median eminence of the hypothalamus, whereas the second set is located in the adenohypophysis. It is the sinusoids of the pars anterior or adenohypophysis. There is the diagram to show the hypophyseal portal system. You can see the red colored circle first set of capillaries, and the blue colored circle second set of capillaries. Next we will discuss neurohypophysis. The neurohypophysis, it does not contain any secretory cells. So the part which contains secretory cells, it is called as adenohypophysis, and the part which contains nervous component is called as neurohypophysis. These two are having different development also, but it is not a part of this lecture. We will discuss in the embryology lecture. So the neurohypophysis, it is mainly composed of unmyelinated axons. These axons, which are coming from the neurons of supraoptic and paraventricular nucleus of hypothalamus. So what are these? Again we will discuss. These are unmyelinated axons. So axons, they arise from the neuronal cell bodies. So these neurons, where they are situated in the hypothalamus, and there are nuclei in the hypothalamus, supraoptic and paraventricular nuclei. So in this diagram we can see the supraoptic and paraventricular nuclei and the axons arising from it, which are entering into the pars posterior. In your short note of pituitary, there should be mention of herring bodies in the neurohypophysis. So herring bodies, what are these? These are swollen axon terminals. These are swollen axon terminals which stores the hormones, that is oxytocin and vasopressin. Vasopressin, it is also known as antidiuretic hormone. The neurohypophysis, it consists of herring bodies. So it can be asked as a multiple choice question. Herring bodies are found in These are found in neurohypophysis. And what are herring bodies? Herring bodies are swollen axon terminals which store oxytocin and vasopressin. Highly branched glial cells. These are called as pituocytes. Highly branched glial cells. These are called pituocytes. Pituocytes. These are present in the neurohypophysis. This could be your Viva question. Pituocytes. What are pituocytes? These are branched glial cells, and where they are present in the neurohypophysis. So what to draw? We have to draw diagram of pituitary gland. Let us see how to draw the diagram. So this is the diagram you have to draw. Now we will discuss how to draw this diagram. So let us see. We will start with the drawing of pars anterior. Pars anterior, we know there are secretory cells. These are named as chromophils and chromophores, and in the chromophils we have to show acidophils and basophils. So we will draw pink colored acidophils, some blue colored basophils, and some cells which are less stained or without stain. These will be our chromophores. So we have shown chromophils and chromophores. So these are chromophores and chromophils. You can see here pink colored acidophils, blue colored basophils, and without stain chromophores. I have also drawn few pigments in each of the acidophil and basophil. Complete the section drawing multiple acidophils and basophils. You need not draw many, but you can show within a part of the slide. Then start drawing capillaries. So there are capillaries which are present between the cells, or we can say in other words, in between the capillaries the cells are present. We have shown the capillaries in the pars anterior. Next we will draw pars intermedia. The characteristic we have already discussed. It consists of colloid filled follicles which are lined by basophilic cells. They are lined by basophilic cells. So let us show colloid filled follicles. Next we will show the axon terminals in the neurohypophysis. Then there will be again capillaries, then glial cells which are pituocytes, the glial cells which are known as pituocytes, and we will show some swollen axons. These are herring bodies. So this is pars anterior, pars intermedia, and pars posterior or neurohypophysis. So this is a section which is sufficient if you draw in the short note, because it consumes very much time. So all the three parts that are seen from the large diagram that we have seen, the bigger diagram that we have drawn, I have cropped a section of this diagram, and all the three parts, that is in anterior part we can see the pars anterior, some colloid filled follicles in the pars intermedia, and in the posterior part we can show some axon terminals, capillaries, and the herring bodies. So if you label properly and draw a smaller section, it is also sufficient to get or to score good marks. So you can draw this much section also out of the entire slide. Next we will discuss the histology of thyroid gland. Again the pattern will be same. It consists of stroma, which is made up of connective tissue, and capillaries. The parenchyma consisting of thyroid follicles, which is the secretory portion of the gland. The blood vessels, again, these are capillaries. Thyroid stroma, it consists of capsule and interlobular septae, which are formed by the extensions of the capsule. So it is loose areolar connective tissue. The capsule, it sends septae dividing the gland into different lobules. So on the histologically as well as the macroscopically or gross anatomy, we can see the lobules in the thyroid gland. What does parenchyma consists? Parenchyma, it shows presence of thyroid follicles. The thyroid follicles, these are structural and functional units of the gland. These are spherical in shape and these will be lined by cuboidal epithelium, simple cuboidal epithelium. So the cells lining the follicles, these are known as follicular cells, which are lined by the follicular cells are simple cuboidal epithelium. These cells line the follicles. So the height of follicular cells, it varies with the secretory activity of the gland. So this is an important statement. Height of follicular cells, it varies with the secretory activity of the gland. So the follicular cells, they synthesize and secrete thyroid hormones. Because this is a part of parenchyma, it will be secreting the hormone. The follicular cells, they secrete thyroid hormones. What is colloid? It is a homogeneous gel-like substance which is present inside the lumen of the follicle. So the hormone, it is stored in this gel-like substance. It is called as colloid or also known as thyroglobulin. Thyroid parenchyma, it also contains parafollicular cells or clear cells, which are also known as C cells. So parafollicular cells or clear cells or C cells, these are large pale staining cells with granular cytoplasm. They found singly or in the group between the follicles, or we can say interfollicular space. The secretion of these cells is calcitonin. The calcitonin is responsible for decreasing the serum calcium levels. That means they decrease the calcium in the blood. The calcium levels in the blood, it is going to decrease. So what we have to draw? We have to draw a basic diagram of the thyroid gland. Let us see. This is the diagram we have to draw for the thyroid gland. Let us see how to draw this diagram. We will begin with a circle. We will start drawing the connective tissue septa, which are dividing the gland into various lobules, and we will draw circular follicles. Draw 3 or 4 circular follicles and show the lining of them, that is a simple cuboidal epithelium lining. Complete the nuclei. The cuboidal cells, they will have a rounded nucleus. Now show colloid or thyroglobulin filled inside each follicle. Draw few more follicles. Complete the diagram. Now show the parafollicular cells between the follicles. These parafollicular cells, they are scattered throughout the gland in between the follicles. They will be either isolated or they will be clustered. So the circular parafollicular cells and the remaining part, that is the capillaries. So draw capillaries and complete the diagram. Again, as we have already discussed, need not draw a complete diagram because it will take much more time. So draw the basic things, that is connective tissue septa, draw few follicles, show parafollicular cells and capillaries. So this will be sufficient to score good marks. So depending upon the time available, you can modify the diagrams. So this is the diagram of active follicle and a resting follicle. So you may draw these diagrams to get few additional marks in your exam. Next we will discuss parathyroid gland. The gland is very small in size, also less frequently asked in the histology short notes, because there are other important glands like pituitary, thyroid and adrenal gland. So if any question asked on the parathyroid, most likely it will be a MCQ, and if it is asked, it will be a odd question. It will be an odd question. Let us see the parathyroid gland. The pattern will be same: stroma, parenchyma and capillaries. The stroma, it is consisting of connective tissue with the capillaries, and the capsule, it will be sending the trabeculae or the septae. and it will merge with the reticular fibers within the gland. Parenchyma, it shows secretory endocrine cells. There are two types of cells. What are these two types of cells? These are the chief cells or principal cells, and another cells are auxiphyal cells. So we will see in the next slide why the chief cells are called chief cells or principal cells. The chief cells, these are smaller and more numerous, and these are secreting parathyroid hormone. So the cells which are serving the function of the gland, that is parathyroid gland, which secretes the parathyroid hormone. This hormone is secreted by chief cells, and these are more numerous. That is why these cells are called as chief cells. So what is the shape of these cells? These are polygonal with round vesicular nucleus, and these are pale staining cells with granular cytoplasm. What is the role of the parathyroid gland? It is opposite to the C cells, parafollicular C cells. The parafollicular C cells, they will decrease the serum calcium, whereas parathyroid hormone, it will increase serum calcium. So it will remove the calcium from the bones. It will remove calcium from the bones, and it will pour it into the circulation. Another type of cells, these are auxiphyal cells. These are large and less numerous, and their function is not known. These are cells with highly acidophilic cytoplasm, that means dark pink color cells. So this is the diagram of parathyroid gland you have to show. So in the parathyroid gland, there are some follicles of thyroid. They have been shown because the gland, parathyroid gland itself, it is embedded in the posterior aspect of the thyroid gland. So in the diagram, they have shown both thyroid as well as parathyroid. Now we will discuss adrenal or suprarenal glands. The gland, it is having peripheral cortex and central medulla. The discussion of suprarenal gland, it is incomplete without the discussion of its development. It is very commonly asked or frequently asked question, development of the suprarenal gland. There are very important questions that have been asked in the development of the suprarenal gland. The cortex, it is mesodermal in origin, whereas medulla, it is neural crest cell origin, that is ectodermal in origin. The cells of the medulla, these are considered to be modified postganglionic sympathetic neurons, which lose their processes and become secretory cells. So what are the cells of medulla? The cells of medulla are modified postganglionic sympathetic neurons. So they will be in contact with the preganglionic sympathetic neurons, which are coming into the medulla of the adrenal glands. So again, I am highlighting this. The cells of the medulla, these are considered as modified postganglionic sympathetic neurons, which lose their processes and become secretory cells. Basically, the cells of adrenal medulla, these are nothing but the gland is covered with capsule like any other endocrine gland. It shows presence of subcapsular plexus, which is formed by the suprarenal arteries beneath the capsule. Parenchyma, it has got two sets of arteries, one set of artery for the cortex and another set of artery for the medulla. The interesting fact about the medulla of the adrenal gland is that it receives the blood, that is the venous blood from the cortex, as well as it has got the arterial blood of its own medulla. So it is exposed to the cortical hormones also. The medulla, it is exposed to the cortical hormones also. So focus on the last statement of the slide. It is cells of the medulla are bathed in both arterial blood from the medullary arteries as well as the venous blood from the cortex. This is very important statement, and this can be asked in the Viva. What is the peculiarity of the medulla of adrenal gland? We know two peculiarities now. Cortex, it consists of zona glomerulosa, zona fasciculata, and zona reticularis. These names are from superficial to deep. Zona glomerulosa, zona fasciculata, and zona reticularis. Zona glomerulosa, it consists of 15% of the total volume of the gland, and it shows presence of rounded or arched clusters, or we can say umbrella shaped clusters of the columnar or pyramidal cells. The cells are acidophilic, that means they are pink in color, and they show presence of lipid droplets. Few lipid droplets are present, and as usual, any endocrine gland, it will show presence of capillaries. So there are fenestrated capillaries found in the zona glomerulosa also. Zona fasciculata, the fasciculus, it means a band. So the zona fasciculata, it shows the bands of the cells. It is the largest part of the gland. It is about 65% of the total volume of the gland. It shows presence of straight parallel cords either in single line or double line fashion. So it is either one cell or two cell thick. The cells, they show pale staining cytoplasm and many lipid droplets, and due to the many lipid droplets, the cells, they are having sponge like appearance. So these are called as spongiocytes. So this could be your MCQ. Spongiocytes, these are found in which gland? Spongiocytes, these are found in adrenal gland. And where exactly in the adrenal gland? The spongiocytes are found in the zona fasciculata of the adrenal gland. And again, there will be fenestrated capillaries. Zona reticularis, as the word reticularis suggests, it is a reticulum or the network of irregularly placed cords of the cells. It consists of 5 to 10% of the total volume, and small irregular anastomosing cords of the cells are present, as we already discussed. The cells are smaller and more acidophilic and separated by the capillaries, fenestrated capillaries. What are the secretions of the zona glomerulosa? These are mineralocorticoids, it is aldosterone. Zona fasciculata, it is glucocorticoids, and zona reticularis, it is small amount of androgen and glucocorticoids again. So zona glomerulosa, mineralocorticoids, zona fasciculata, glucocorticoids, and zona reticularis, it is sex steroids or androgens in few amount. Now let us discuss the adrenal medulla. As we already know, the adrenal medulla, it shows the modified postganglionic sympathetic cells or sympathetic neurons which have lost their axons. So these cells, these are known as chromaffin cells or pheochromocytes because they are having affinity for the chromic salts. So why these are called as chromaffin cells? These are having affinity for the chromic salts. Fenestrated capillaries will be there. There will be fine reticular connective tissue. So what is chromaffin reaction? The cells stained with the chromium salts. It is called as chromaffin reaction. So the preganglionic sympathetic neurons reaching into the medulla, these are cholinergic nerve endings. These are cholinergic nerve endings. They secrete catecholamines like epinephrine and norepinephrine, which are also known as adrenaline and noradrenaline. There are also few parasympathetic ganglion also present. So this is the diagram you should draw, and how we should draw this diagram, that we will see in the next frames. Let us start with drawing the capsule. We will draw capsule first. Beneath the capsule, we will show presence of circular clusters of the cells or arched clusters of the cells, which is our zona glomerulosa, presence of glomeruli, the secretory cells. So these are clusters of the cells, arched clusters, zona glomerulosa, and we will show some sinusoids or fenestrated capillaries also. Now, in the next frame, we will show presence of cords of the regular cords. These are zona fasciculata. These are zona fasciculata. These single or double layered cords of the cells which are separated by the sinusoids, they form zona fasciculata. Complete the diagram. Then show zona reticularis, the irregularly arranged cords of the secretory cells which are separated by the capillaries. Once you complete the drawing of zona reticularis, start with the drawing of medulla. In the medulla, you will show the cells, that is chromaffin cells, which are having affinity for the chromic salts, and we will show the sinusoids. This is medulla, and above this is cortex. So you can draw this much diagram also. If the time is short, you can show this much portion of the diagram also. So all the labels are covered in this much portion. So capsule, zona glomerulosa, then we have zona fasciculata, zona reticularis, and we have already shown the medulla with medullary veins and the chromaffin cells. Sometimes in Viva, you may get a question, what is the difference between adrenal cortex and adrenal medulla? Few of the important differences we already discussed. Now we'll discuss few more. The cortex is under control of the hormones from the pituitary, that is adrenocorticotrophic hormone. Cortex is under control of adrenocorticotrophic hormone. We have already discussed pituitary. We know it is secreted by the pars anterior. Then cells of the cortex, they do not store their hormones in the granules, whereas the medulla, it is under neuronal control, because we already discussed these are modified postganglionic sympathetic neurons. The cells of the medulla, they accumulate and store their secretory products in the granules. Next, the less important gland from the theory point of view is pineal gland, which is also known as epiphysis. There are two types of cells. One are called as pinealocytes. They form the bulk of the gland. These are modified neurons. What are pinealocytes? These are modified neurons. They are about 95%. The pinealocytes, they have long tortuous cytoplasmic processes ending in relation to the blood vessels as swellings, and these swellings, they store the granules containing melatonin and its precursor serotonin. So the melatonin is the secretion of pineal body. You should prepare this multiple choice question. The secretion of pinealocytes or the secretion of pineal gland is melatonin. Next, it is the supporting framework by the astroglial cells, which are also known as interstitial cells. The pineal gland basic structure, it is same, pods and clusters of cells separated by the capillaries. The important multiple choice question that is asked in the pineal gland topic, it is presence of corpora arenacea, which is also known as brain sand. So these are basically basophilic bodies which are found in the pineal gland extracellularly within the interstitium of the pineal gland. The brain sand is nothing but the concentric layers of calcium and magnesium phosphate which accumulate in the pineal gland. So as they contain calcium, they serve as an important landmark of the pineal gland in the X-ray skull. This is the diagram of the pineal gland that you should draw. I haven't made a how to draw session of the pineal gland as well as parathyroid gland. So it is advised that students should practice them from the textbook. We have covered pituitary, thyroid and adrenal, three important endocrine glands in this topic. So that is all from endocrine glands. For more sessions like this, don't forget to subscribe the channel, that is Easy Anatomy for UG. Share it with your friends, ask them to subscribe and follow, and don't forget to comment on this video whether it was useful or not. I will see you with one more easy session of anatomy. Thank you.